Alright guys, we're back to finish up Chapter 7 with Concept 7.4. I said I was going to get some iced tea and I lied. I went and got Dr. Pepper instead. Sugar and caffeine, two of the most important food groups for every single teacher on the planet. Alright, so Concept 7.4 says, active transport uses energy to move solutes against their gradients. We had already talked about passive transport, which was moving materials down their gradients from an area of high concentration to an area of low concentration. But for cells to maintain life and to do all the various things they have to do, sometimes they have to actually move materials against their concentration gradient. In other words, they have to take material and move it from low concentration to an area of higher concentration. And this is going against the grain. This is going against this equilibrium that nature is usually after. So this almost always requires energy, and this is called active transport. So it requires some input of energy on behalf of the cell. We talked about facilitated diffusion, how that uses the proteins, either carrier proteins or channel proteins. That's still passive because the material is moving down the concentration gradient. It helps speed things along, but it doesn't really affect the direction. That material was going to go that way anyway. So, for example, if we're talking about water and there's a larger concentration of solutes inside the cell, water was going to be moving into the cell anyway. The aquaporids just help speed up that process. But sometimes we have to go the opposite direction. We have to go against the natural flow, against the normal flow. We have to move up our concentration gradient from a lower to a higher concentration, and that's going to require energy. So we call that active transport. Because we're moving against the concentration gradient, we are going to have to use energy to do this. The vast majority of the time, energy in cell terminology is going to be the ATP molecule we talked about, the adenosine triphosphate. So active transport is performed by some of these proteins that we discussed already that was embedded in the membrane. And these are all going to be carrier proteins. So there are no channel proteins that will work against the gradient, against the concentration gradient, because all the channel proteins do is provide a path or a tunnel. So that's only going to be diffusion. If we're talking about going against the current, against the diffusion gradient, then we're going to have to use carrier proteins, no channel proteins. So active transport allows cells to maintain a concentration gradient that's different from their surroundings. In other words, this allows the cells to maintain an imbalance that is desirable or an imbalance that is necessary. And to do that, they have to have some sort of, almost like a pump system of some way. And one of the carrier proteins that's most well known is called the sodium-potassium pump. It's one type of active transport system. In animal cells, when you compare the inside of the animal cell to its outside environment, the inside requires a higher concentration of potassium ions and a lower concentration of sodium ions. So to keep this uneven concentration inside and outside the cell, we have to use active transport in this sodium-potassium pump. Great little animation right here. So if we just look at this picture, we can see kind of what's going to happen. And then if you right-click and hit play, you can see the actual animation. The blue area here is outside the cell. The tan area is inside the cell. And I want you to notice that there is material swapping into and out of the cell, and both of these are positive ions. But I want you to notice very carefully that this is not an even swap of positive ions in and out. Every time this pump runs, every crank of the pump, you could call it, that sends 3 positive sodium ions out of the cell and brings in 2 positive potassium ions. So that's not an even amount or not an even number. Over time, this makes the extracellular environment out here more positive than the intracellular environment, the intercellular environment. So what that does is it causes an imbalance where we have more positive charge outside of the cell than inside the cell. The cell needs this, and we'll talk about that in just a moment as to why, but this increases what we call membrane potential. You're building up electrical potential energy because the charge difference outside the cell doesn't match what's inside the cell. So hit right click and play and take a look. So if you didn't get a chance to see that animation or you just wanted a little bit further explanation, here it is broken down into about 6 little steps. So this is the sodium-potassium pump. a very important carrier protein, the transport protein found in animal cells. So let's start with one right here, and we'll move our way to the right and then down and to the left. So here at stage 1, we have sodium positive ions coming from inside the cell, and they are latching on to the carrier protein. When they get 3 latched on, there is an ATP molecule that attaches that provides energy. That energy allows this protein molecule to change its shape and release the sodium ions to the outside of the cell. Then, once that occurs, positive potassium ions, 2 of them, can come from outside the cell, attached to the protein. Then the ATP is released, the used ATP basically is released. That changes the shape of the protein molecule again and allows the potassium ions to be released inside the cell. So this ratcheting, this changing of shape, takes an ATP molecule to cause that to happen. It requires energy. And you notice 3 positive ions are leaving the cell, 2 positive ions are entering. So in effect, every run of the pump causes 1 extra positive ion to be outside of the cell than inside. And that causes an increase in what we call membrane potential. And basically now, since we're talking about charges inside and outside the cell, we're creating a battery where there is a difference in charge on one side than the other. That allows for potential energy, and that potential energy can then be used to do other work that we're going to talk about later. So before we go on, let's do a really quick review. I love this picture right here. It's also in your textbook, figure 7.19. It's a review of passive and active transport. So first we have on the left all sorts of passive transport. Remember, that goes down the concentration gradient from higher concentration to lower concentration of solute. Does not require energy from the cell. So the first one is just straight up diffusion, where the material moves across the lipid bilayer, the phospholipid bilayer, into the cell all by itself. The other 2 over here are called facilitated diffusion. They're still diffusion. They're still moving down the concentration gradient with no energy from the cell, but they're helped along by proteins, transport proteins. Here we have a channel protein that basically allows for a tunnel or a pathway to travel into the cell. Probably the most well-known is the aquaporin that allows water to come in. And then here we have a carrier protein. The carrier protein changes shape, and that allows the material to pass through, but because it's still going with the concentration gradient, it doesn't require an ATP molecule or energy input from the cell. Over here on the right, we have active transport. Active transport does require energy, and it's moving material against the concentration gradient. So from a low concentration to a higher concentration. So these little square molecules, see, they're moving from low concentration to higher concentration within the cell. That can only be done with the input of energy in the form of ATP. Probably the most well-known active transport protein or system is the sodium-potassium pump in the animal cells. And so let's talk about that membrane potential. That sodium-potassium pump builds up positive ions on the outside of the cell. So the inside is more negative, the outside is more positive. You're basically creating a battery that has potential energy stored that can be used to do other things. So let's discuss briefly what that membrane potential is. It's basically the voltage difference across a membrane, and that's going to be the charges, positive or negative. If they're not equal on either side of the membrane, you've set up a voltage difference which gives you what we call membrane potential. The inside of a cell is generally negative compared to more positive on the outside. And this voltage difference is created by that imbalance of positive and negative charges. So, and again, I think I said it previously, you're basically creating a battery where one side is more negative or positive than the other, and that allows for the storage of potential energy that can be used for other jobs. So in effect, we have 2 different things that drive the process of moving materials back and forth across the membrane. Combined together, we call these the electrochemical gradient. The electrochemical gradient. And that is what drives the diffusion of ions, charged particles across a membrane. If we're talking about just neutral molecules or neutral materials, we don't really involve the electricity. But if we're talking about ions, which are negative or positive charged particles, well, we talk about this whole electrochemical gradient. We've got the chemical part, which is the concentration gradient, And then we have the electrical part, which is the difference between the positive and negative charges, the membrane potential on the ion's movement. Sometimes these forces work with each other. Sometimes they actually work against each other. But if you're talking about moving ions that have a charge, you have to take into account two things. You have to take into account the concentration gradient and then the membrane potential, because both of those things help either push material in or out of the cell. And remember, positive cations want to move into the cell because the inner part of the cell tends to be more negative, and negative anions tend to want to move out of the cell because outside of the cell tends to be more positive, and they're trying to reach that equilibrium or balance. The sodium-potassium transport protein we talked about, the sodium-potassium pump, is also called an electrogenic pump. An electrogenic pump is one of these transport proteins that as it does its job, it's building up a voltage across the membrane. So the sodium-potassium pump, another name for that is an electrogenic pump in animal cells, because as it does its job, it further builds up this membrane potential because it creates a larger and larger difference in the charge inside the cell as opposed to outside the cell. The main electrogenic pump in plants and fungi and bacteria is not the sodium-potassium pump. It's a different kind of pump called a proton pump. And it's called a proton pump because it's moving protons, which are basically just H plus hydrogen ions without their electron. It's moving H plus hydrogen ions out of the cell. And that would be against the concentration gradient because it's already more positive outside the cell, and this is forcing more positive ions outside the cell. So electrogenic pumps, doesn't matter if it's a sodium-potassium pump in plants, sodium-potassium pump in animals, or a proton pump in plants or fungi or bacteria. All these electrogenic pumps, as they do their job, they're helping store energy that can be used for work later on by building up this membrane potential. Here's a great little example, picture here of a proton pump. That would be one of these electrogenic pumps in a plant cell or a fungi cell, and it's taking hydrogen ions, has to use ATP to do this, and it's forcing that hydrogen ion out of the cell where there's already a higher concentration. So outside of the cell, you're building up a positive charge. Inside the cell, it becomes more negative. Just to give you a couple of examples of, so the plant is building up this charge, or the animal cell is building up this charge. What is it going to use that charge for? What is it going to use that stored potential energy for? Well, we're going to find out there's actually a couple of different reasons it might use this energy. One of them is to create ATP synthesis during a process called cellular respiration, which of course we'll look at a little bit further down the line. And then the other one is what we're going to call co-transport of materials. So the cell may use this energy difference to get some work done. One of those jobs may be to create more ATP, and that's called cellular respiration. Again, we'll look at that in detail later in the semester. And the other one that we're going to look at today is called co-transport of materials. So co-transport is kind of like a multi-step process. It's kind of a more lengthy, complicated way of moving material across a cell membrane. And co-transport happens when the active transport of a material indirectly drives the transport of another material. So the active transport of moving one product in and out also allows another one to kind of piggyback or hitchhike along and come in and out as well. A great example is in the plant cells we were talking about with that hydrogen proton pump. So they use this proton pump to pump hydrogen ions out of the cell and make it more positive on the outside and more negative on the inside. Well then, remember, those hydrogen ions really want to just rush right back in. They want to try to even out that charge, and it's more negative inside the cell, so the positive hydrogen ions want to get in there. So they can diffuse all by themselves through the cell membrane, or they can come through, and as they come through, that work, that energy can be used to move something else into the cell as well. So imagine you have a water pump and you have a big hill and you got a lake at the bottom of the hill. You're using a water pump, an electric water pump or a diesel motor water pump. You're pumping water up the hill, and then you're letting that water come out of the hose way at the top of the hill, and you're letting it run down the hill naturally all by itself. But in the middle of the hill, you got a big paddle wheel, like a big wheelhouse that you might see for grinding corn or grinding grain that they used to use hundreds of years ago. So you've got a big water wheel, a big paddle wheel, and as that water naturally flows back down the hill, it's turning this paddle wheel that allows you to grind the corn or do some work or something like that. So like pumping water up a hill and then using the natural flow of the water back down to turn a wheel and get some work done, that's kind of like co-transport or coupled transport. So we'll take a look at a picture perhaps in just one second. So here we have a model of a plant cell. Now they've taken out the cell wall just to make the model cleaner, so you just see the cell membrane, but this is a plant cell, or it could be a fungi cell, something like that. So we have the proton pump right here. Remember, that's active transport. It's an electrogenic pump. It is using ATP to pump out hydrogen ions against the concentration gradient, making it more and more positive outside of the cell. Well, those ions, they really want to just diffuse right back in to help even this electrical gradient out. So they start to diffuse back in. Well, that diffusion back in allows this co-transporter protein to take sucrose and move it into the cell. Sucrose by itself would have a very hard time moving through the lipid bilayer. It would take a long time and it would be a very slow process. Sucrose is a larger molecule, and it's also hydrophilic, so it would have a hard time getting through the lipid bilayer. So here we have the natural diffusion of the hydrogen ions back in, allowing to basically sneak sucrose across using this protein right here. That's co-transport. So I guess the real question down here is, why would the cell do this? Co-transport seems kind of like a two-step method where you could just do straight-up active transport to begin with. Just have a protein that pumps sucrose across using an ATP. Well, the reason for that is probably because evolution is a messy process. Evolution rarely creates perfect systems. There are many body systems and cellular systems where you look at and you think, well, yeah, it works, but man, it really could work so much better if these two steps were taken out, or it could be a lot cleaner and more simple. Well, the problem is evolution doesn't work toward a perfect system. Evolution works toward a working system, and it's a working system that had to be cobbled together using what already existed. So the cell, to create this system, had to use existing parts and other existing systems and perhaps co-opted another system. So you'll find this oftentimes when we start to talk about anatomy and physiology and all sorts of variations of species. You'll find that in many cases it works, but man, it could be a lot cleaner and simpler and easier. But that's not the way that evolution works. Evolution has to take what it's got and make do, and as long as it makes do well enough to work, that's probably all you're going to get. Alright, last concept, gang, concept 7.5. What if we need to move a lot of material, or we need to move big molecules in and out of the cell? Well, we're probably not going to be able to use any of the methods we already talked about. Diffusing through the cell membrane would be very difficult for a large, complex molecule. Probably difficult to use any of the carrier proteins. You know, it would be difficult to find a transport protein that's large enough to lock on and allow it to pass through, or a channel protein that'll allow that. So if we're going to move large amounts of material or very large complex molecules, we're going to use what's called exo and endocytosis. So concept 7.5 says bulk transport across the plasma membrane occurs by exocytosis and endocytosis. If you're a little molecule like water or some other small molecule, you can go in and out of the cell right through the cell membrane or through some transport protein. But if you're a big molecule, some polysaccharide or some large protein, you're going to have to go in a vesicle. And remember, we already talked about the vesicles, how the vesicles are basically just pinched off parts of the endoplasmic reticulum and the Golgi apparatus, and they are like transport cargo vans that can move large amounts of material. And of course, just like a cargo van requires gasoline, to move a large molecule or a large chunk of molecules with a vesicle, that's going to require energy. So let's start with exocytosis. You can probably guess from the name that exocytosis is moving materials out of the cell. In exocytosis, the transport vesicle will eventually fuse with the outer plasma membrane and release whatever it was carrying out into the cell. And most of our what are called secretory or secretory cells use this method to export their product. So a great example is like in the pancreas. Your pancreas produces a chemical called insulin. It's a material called insulin that helps regulate the sugar balance in your blood. So the pancreas, the cells produce insulin. That insulin is packaged in vesicles that fuse with the cell membrane, and then that insulin is released to the outside of the cell into the extracellular fluid, where eventually it travels and takes its way into the blood and all the other parts of the body. That's exocytosis, leaving the cell. Great little quick animation of exocytosis. If you just hit right click and play, and you can see what's going on. The opposite of that, of course, is endocytosis. Think about coming in. Of course, it's not spelled the same, but endocytosis, so that would be your entry point coming in. Endocytosis is where big macromolecules are taken in by the cell. The cell membrane sort of forms a little vesicle around and captures the large molecule and then comes in, pulls it into the cell. And there's really 3 different types of endocytosis. We've actually kind of talked about them before. One of them is phagocytosis or phagocytosis, and that is eating. That's like how an amoeba eats its food. It's also how large molecules are brought into the cell. Phagocytosis or phagocytosis. And think about that as eating, cellular eating. The other one is penocytosis or pinocytosis. Pinocytosis is drinking. This is kind of like where the cell basically just takes a gulp of the extracellular liquid around it, and in that gulp is going to be a lot of different materials, but including some of the materials that the cell needs or wants. And then the last one's a little bit fancier. It's called receptor-mediated endocytosis, and that's kind of like fishing, where the cell is looking for some particular material, and it's going to try to attract that material to this site where it forms a vesicle around it and then pulls it inside. Here's another quick little video. Kind of gives you a quick overview of both exocytosis and endocytosis. If you just want to right click and hit play, you should be able to see that. So let's take a look at those 3 different types of endocytosis. The first one is phagocytosis or phagocytosis. That's where a cell engulfs the particle that it wants and then creates a vesicle around that to pull it into the cell. And then the vacuole that gets pulled in, it could be a food vacuole or some other sort of transport vacuole that gets brought into the cell. Then the lysosome, remember the lysosomes are the digestive vesicles. The lysosome will then attach to it and open up and digest whatever was brought in. So here we have some kind of microscope pictures, electron microscope, scanning microscope pictures of phagocytosis going on, where the green cell is basically ingesting this other cell or this other large molecule, probably another cell based on the size. Quick little animation of phagocytosis. If you just want to hit right click and play, you can see that. The second one we mentioned was penocytosis or penocytosis. Penocytosis is basically drinking. So the cell is basically taking a gulp of the liquid that surrounds it, and in the process it's taking up some of the molecules that it needs out of that extracellular fluid. So here's a quick little animation of penocytosis. Again, right click and play. The last one was the one that was a little bit fancier type of endocytosis, and it's called receptor-mediated endocytosis. So basically what happens is the cell is looking or searching, kind of fishing for particular molecules. And what it has on the outside of the cell are particular receptor sites, and it's looking for what's called a ligand. A ligand is a molecule that specifically binds to a particular site. So you can think of the receptor site on the outside of the molecule as the fishing lure, and the ligand is kind of the fish that it's going after. So that ligand is just a general name for any molecule that binds to a specific receptor site. So the cell will have receptor sites on the outside of the cell membrane, and those are basically fishing poles, and they are all set up for one particular fish, one particular ligand. And it will probably generally line up a bunch of those in one area. So there'll be a lot of these particular binding sites, receptor sites in one area of the cell membrane, and it will capture a number of these ligands and then form a membrane around it and pull it inside. Now it'll usually capture other material as well, but there'll be a higher concentration of what it's looking for. So maybe not thinking about it as a specific fishing lure for one specific type of fish, but maybe a particular type of fishing net that will catch, for the most part, the fish that it wants, but also a few extra as well. So here's a quick animation of the receptor-mediated endocytosis. This one's probably particularly important to watch because that one's a little bit more confusing. So it is endocytosis, but it's kind of targeted endocytosis. Great overview on page 139 of your textbook. Here you have all of the three different types of endocytosis, the phagocytosis, where it brings in a large molecule, forms a vesicle around it, and then later on the lysosome will fuse with this vacuole and then digest that material. Here we have the pinocytosis, where it's kind of taking a gulp of the fluid and forming a vesicle, pinching off a vesicle, bringing that inside. And then receptor-mediated endocytosis is where it has these particular receptors, the little red V or Y-shaped. Those are the receptors, kind of like the fishing lures. And then it's looking for a particular ligand, and the ligand here is this purple triangle. So it gets those ligands attached and then forms a vesicle around and pinches it off and brings it inside. Notice there's more than just the purple triangles it was looking for, but it's still pretty good high concentration of what it was needing. Another quick shot showing not just the model, but also, I love it when they show the real microscope view. Here's an amoeba eating a bacteria. This amoeba is not much larger than the bacteria, but he is basically surrounding the bacteria and ingesting it, bringing it in via phagocytosis or phagocytosis. The pseudopodium of the amoeba, which is the false foot, which is about to encircle that, bring it in and form a vesicle around it. Here's the pinocytosis. Again, you see these little vesicles forming where the little arrows are, taking gulps of that liquid and bringing it inside. And here's receptor-mediated endocytosis. The little proteins, you can see these little projections sticking out. Those are the receptor sites, and it's binding with certain ligands, and then forming this round vesicle around them, pinching off and bringing that inside. Cool shot with a TEM, tunneling or transmission electron microscope. So we'll finish right there. All sorts of ways in which the cell gets products in and out of itself. Some of them are passive ways that don't require energy, like diffusion and osmosis, which is diffusion of water. Some of them use transport proteins, but it's still down the concentration gradient, so it would still be without using energy, still be passive. And then other ways require energy. So if you're moving up the concentration gradient, you're going to require energy. That's going to require active transport, and it's going to require the use of an ATP molecule. Lots of different ways to do that. And then this last part was how to move big, bulky materials or lots of materials in and out, and that would be using vesicles. We're going to stop right there. Check out the crash course video called In the Club. Hank's got a good, entertaining take on all of this. There's a link right there. I'll see you guys next time around for Chapter 8. Just as a quick heads up, the next chapters are all about, so how do the cells get energy to do all this? I mean, we keep saying that it requires energy to do some of these things. How do the cells get that energy? Well, that's what's coming up next. See you guys.